Hi everyone. In this lab, we're going to be looking at the effect of temperature on enzyme action, and we're going to be using a reaction where hydrogen peroxide goes through a decomposition reaction to break down into water and oxygen. Hydrogen peroxide is a toxic byproduct of metabolism, and if the cell does not break it down quickly, it is going to kill the cell. So hydrogen peroxide has an enzyme to help it to do this quickly, and that is called catalase. Hydrogen peroxide is the substrate that we're going to be using. It is toxic to cells, and so cells make an enzyme called catalase that will break it down. So for our catalase for this reaction, we're going to be using yeast, and I have hydrogen peroxide in both the beaker and the bowl, and the bowl is there so that you can see the reaction. So I'm going to pour this in so that you can see what happens, and you can see that it becomes all bubbly. Right, you can see the bubbles coming up. Now we're going to utilize this reaction with the bubbles to float a paper disc. So theoretically, if the paper disc is in there, it's going to, the oxygen bubbles are going to attach to the paper disc and carry it to the top. So here I have hydrogen peroxide, and in this lab, what we would do is we would take this little paper disc and we would dip it into the yeast solution. And what we're going to do is we're going to drop it down, and you can see that it's down at the bottom and it floats up to the top when there are enough bubbles there to carry it up. And what the scientist would do is measure the time for it to, from the moment it goes in to the moment it hits the top. And we're going to be testing temperature with this by changing the temperature of the hydrogen peroxide. So to clarify, the first thing that the scientist would do would be to measure the temperature of the hydrogen peroxide that had been placed in the beaker. They would use a pair of forceps to take a paper disc and soak it in yeast and be ready to start timing. As soon as the paper disc was dropped, time would start. Now, as soon as the paper disc drops down, gravity pulls it down to the bottom. But remember that it's covered with yeast, and this is hydrogen peroxide right here. So when the catalase from the yeast interacts with the hydrogen peroxide, we start to see a breakdown, and we start to see oxygen bubbles produced. Now, those oxygen bubbles are going to cling to the paper to start. When there's enough oxygen on the disc is going to begin to rise up to the top, and when it reaches the top, that is when time will stop. The time it takes for the paper to gather enough oxygen to reach the top is an indication of how much reaction is actually taking place, and we can compare that between our temperatures. We repeat this 3 times at each temperature, and we have 5 different temperatures worth of data on the lab. You will be averaging your data and graphing your data according to the instructions and finishing up the analysis. When your lab is done, submit the completed document to the assignment on Schoology.